Hello students, welcome to this program. Today's lecture will be on brief introduction to genetics. The main objectives that we are going to cover are to know about the basic science of genetics and to know about the molecular mechanism of genetic information. First, we are going to study introduction to genetics. Genetics is the branch of the biology that deals with heredity and variation. It is concerned primarily with understanding biological properties that are transmitted from parent to offspring. The subject matter of genetics includes heredity, the molecular nature of the genetic material, the ways in which genes, that is, which determine the characteristics of organisms, control life functions, and the distribution and behavior of genes in populations. Genetics is central to biology because gene activity underlies all life processes from cell structure and function to reproduction. It stands as the unifying discipline in biology by allowing us to understand how life can exist at all levels of complexity. Ranging from molecular to the population level, genetic variation is the root of the natural diversity that we observe among members of the same species as well as among different species. Genetics is centered on the study of genes. A gene is classically defined as a unit of heredity. At the molecular level, a gene is a segment of DNA that produces a functional product. The functional product of most genes is a polypeptide, which is a linear sequence of amino acids that folds into units that constitute proteins. Now coming to Mendel's work on transmission of traits. The work of Georg Johann Mendel is considered the foundation of modern genetics. In 1854, he began a series of breeding experiments with garden pea by some sativum to learn something about the mechanisms of heredity. As a result of his creativity, Mendel discovered some fundamental principles of genetics. From the results of crossbreeding pea plants with different traits involving seed shape, seed color, and flower color, Mendel developed a simple theory to explain the transmission of hereditary traits from generation to generation. Mendel reported his conclusions in 1865, but their significance was not fully realized until the late 1800s and early 1900s. Mendel's experimental approach was effective because he made simple interpretations of the ratios of the types of progeny he obtained from his crosses, and because he then carried out direct and convincing experiments to test his hypothesis. In his initial breeding experiments, he took the simplest approach of studying the inheritance of one trait at a time. He made carefully controlled matings, or what we call as crosses, between pea strains that had obvious differences in heritable traits, and most importantly, he kept very careful records of the outcomes of the crosses. The numerical data he obtained enabled him to do a rigorous analysis of the hereditary transmission of traits. Mendel did all his significant genetic experiments with the garden pea. The garden pea was a good choice because it fits many of the criteria that make an organism suitable for use in genetic experiments. It is easy to grow, bears flowers and fruits in the same year a seed is planted, and produces large number of seeds. Mendel obtained 34 strains of pea plant that differed in number of traits. He allowed each strain to self-fertilize for many generations to ensure that the traits he wanted to study were inherited. This preliminary work ensured that Mendel worked only with pea strains in which the strains under investigation remained unchanged from parent to offspring for many generations. Such strains are called true breeding or pure breeding strains. Next, Mendel selected 7 pairs of traits to study in breeding experiments. Each pair affected one characteristic of the plant, with each member of a pair being clearly distinguishable. Flower and seed coat color, gray versus white seed coats and purple versus white flowers. A single gene controls both these color properties of seed coats and flowers. Second is the seed color, yellow versus green. Third, the seed shape, smooth versus wrinkled. Fourth, pod color, green versus yellow. Fifth, pod shape, inflated versus pinched. Sixth, stem height, tall versus short. And finally, flower position, axial versus terminal. Now coming to genetic variation. Genetic variation means when genetic structure of population varies in space and time. This means that the frequencies and distribution of alleles can vary in samples of the same species in different geographic areas or samples from the same geographic area collected at different generation times. Many populations of plants and animals that have widespread geographic distributions show distribution of this sort in the allele frequencies of population in different geographic regions. In some cases, the spatial variation shows clear patterns or trends across geographic space. When allele frequencies change in a systematic way across a geographic transect, we call this an allele frequency cline. Often clines are associated with changes in physical attribute in the environment, such as temperature or water availability. Genetic variation within population is important for several reasons. First, it determines the potential for evolutionary change and adaptation. The amount of variation also gives us important clues about the relative importance of various evolutionary processes, because some processes increase variation while others decrease it. The manner in which new species arise and existing populations become extinct may depend on the amount of genetic variation. How about within populations? In addition, the ability of a population to persist over time can be influenced by how many genetic variation it has to draw by environment's change. Now, the molecular basis of genetic information. The function of genetic material is that of a blueprint. It stores the information necessary to create a living organism. The information is contained within units called genes. At the molecular level, a gene is defined as a segment of DNA that is used to make a functional product, either an RNA molecule or a polypeptide. The first step in this process is called transcription, which literally means the act or process of making a copy. In genetics, this term refers to the process of synthesizing RNA from a DNA sequence. The structure of DNA is not altered as a result of transcription. Rather, the DNA base sequence has only been assessed to make a copy in the form of RNA. Therefore, the same DNA can continue to store information. DNA replication provides a mechanism for copying that information so it can be transmitted from cell to cell and from parent to offspring. Structural genes encode the amino acid sequence of polypeptide. When a structural gene is transcribed, the first product is RNA molecule known as messenger RNA or simply mRNA. The process of polypeptide synthesis is called translation. The sequence of nucleotides within the mRNA determines the sequence of amino acids in a polypeptide. One or more polypeptides then assemble into a functional protein. The synthesis of functional proteins ultimately determines an organism's traits, which is called the central dogma of genetics, or also called the central dogma of molecular biology. It was first demonstrated by Francis Crick in 1958. It forms a cornerstone of our understanding of genetics at the molecular level. The flow of genetic information occurs from DNA to mRNA to polypeptide, which forms functional protein. Now lastly, an overview of Mendel's experiments, or rather we can say explaining Mendel's ratios. In one experiment, Mendel cross-fertilized or simply crossed tall and dwarf pea plants to investigate how height was inherited. He carefully removed the anthers from one variety before its pollen had matured and then applied pollen from the other variety to the stigma, a sticky organ on the top of the pistil that leads to the ovary. The seeds that resulted from these cross fertilizations were sown the next year, yielding hybrids that were uniformly tall. Mendel obtained tall plants regardless of the way he performed the cross, that is, tall male with dwarfed male and dwarfed male with tall female. Thus, the two reciprocal crosses gave the same results. Even more significantly, however, Mendel noted that the dwarf characteristics seemed to have disappeared in the progeny of the cross, for all the hybrid plants were tall. To explore the heredity makeup of these tall hybrids, Mendel allowed them to undergo self-fertilization, the natural course of events in peas. When he examined the progeny, he found that they consisted of both tall and dwarf plants. In fact, among 1064 progeny that Mendel cultivated in garden, 787 were tall and 277 were dwarf, a ratio of approximately 3:1. Mendel was struck by the reappearance of the dwarf characteristic. Clearly, the hybrids that he made by crossing tall and dwarf varieties had the abilities to produce dwarf progenies, even though they themselves were tall. Mendel inferred that these hybrids carried a latent genetic factor for dwarfness, one that was masked by the expression of another factor for tallness. He said that the latent factor was recessive and the expressed factor was dominant. He also inferred that these recessive and dominant factors separated from each other when the hybrid plants reproduced. This enabled him to explain the reappearance of the dwarf characteristics in the next generation. Mendel performed similar experiments to study the inheritance of 6 other traits, that are seed texture, seed color, pod shape, pod color, flower color, and finally the flower position. In each experiment, called a monohybrid cross because a single trait was being studied, Mendel observed that only one of the two contrasting characters appeared in the hybrids, and that when these hybrids were self-fertilized, they produced two types of progeny, each resembling one of the plants in the original crosses. Furthermore, he found that these progeny consistently appeared in a ratio of 3:1. Thus, each trait that Mendel studied seemed to be controlled by a heritable factor that existed in two forms, one dominant and the other recessive. These factors are now called genes, a word coined by the Danish plant breeder William Johnson in 1909. Their dominant and recessive forms are called alleles, from the Greek word meaning of one another. Alleles are alternate forms of a gene. The regular numerical relationship that Mendel observed in these crosses led him to another important conclusion, that genes come in pairs. Mendel proposed that each of the parental strains that he used in his experiments carried two identical copies of gene. In modern terminology, they are diploid and homozygous. However, during the production of gametes, Mendel proposed that these two copies are reduced to one. That is, the gametes that emerge from meiosis carry a single copy of a gene. In modern terminology, they are haploid. Mendel recognized that the diploid gene number would be restored when sperm and egg unite to form a zygote. Furthermore, he understood that if the sperm and egg came from genetically different plants, as they did in his crosses, the hybrid zygote would inherit two different alleles, one from the mother and other from the father. Such an offspring is said to be heterozygous. Mendel realized that the different alleles that are present in the heterozygote must coexist even though one is dominant and the other is recessive, and that each of these alleles would have an equal chance of entering a gamete when the heterozygote reproduces. Furthermore, he realized that random fertilization with a mixed population of gametes, half carrying the dominant allele and half carrying the recessive allele, would produce some zygotes in which both alleles were recessive. Thus, he could explain the reappearance of the recessive characteristic in the progeny of the hybrid plants. Mendel used symbols to represent the heredity factors that he postulated, a methodological breakthrough. With symbols, he could describe hereditary phenomena clearly and concisely, and he could analyze the results of crosses mathematically. He could even make predictions about the outcome of future crosses. The allelic combination of each strain is said to be genotype. By contrast, the physical appearance of each strain, the tall or dwarf characteristic, is said to be its phenotype. Their hybrid progeny are referred to as the first filial or F1 from a Latin word meaning son or daughter. Because each parent contributes equally to its offspring, the genotype of the F1 plants must be capital D and small d, that is, they are heterozygous for the allele of the gene that controls plant height. Their phenotype, however, is the same as that of the D parent strain because D is dominant over small d. During meiosis, the F1 plants produce two kinds of gametes, D and small d, in equal proportions. Neither D is changed by having coexisted with the other in a heterozygous genotype. Rather, they segregate or separate from each other during gamete formation. The process of allele segregation is perhaps the most important discovery that Mendel made. Upon self-fertilization, the two kinds of gametes produced by heterozygote can unite in all possible ways. Thus, they produce four kinds of zygotes. That is D, small d, capital D, and finally small d and small d. However, because of dominance, three of these genotypes have the same phenotype. Thus, in the next generation called the F2, the plants are either tall or dwarf in a ratio of 3:1. Mendel took this analysis one step further. The F2 plants were self-fertilized to produce an F3. All the dwarf F2 plants produced only dwarf offspring, demonstrating they were homozygous for small d allele, but the tall F2 plants comprise two categories. Approximately one-third of them produced only tall offspring, whereas the other two produced a mixture of tall and dwarf offspring. Mendel concluded that the third that were true breeding were D homozygotes, and that the two-thirds that were segregating were D small d heterozygotes. These proportions 1/3 and 2/3 were exactly what his analysis predicted, because among the tall F2 plants, the D small d genotypes occur in the ratio of 1:2. Thank you. This was all from today's lecture. Hope you understood it well. Thank you.